Okay, thank you. So, good afternoon. My name is Jiyu Chen, and I'm currently a research fellow, a postdoc research fellow at the Commonwealth Science and Industry Research Organization from Australia. And thank you for giving me this opportunity to introduce our recent studies on integration of background knowledge for automatic detection of inconsistencies in gene ontology annotation. Oops, how do I get to? Yeah, so firstly, let me begin by introducing what is gene ontology annotation and specifically what is literature-based gene ontology annotation. So a gene ontology annotation is a biological database record produced through a process where experts analyze the scientific literature to manually assign functional information to genes or gene product based on the experimental evidence reported in the research articles. So let's see an example of how a piece of gene function information is captured and stored using the gene ontology annotation. So this is the abstract of a publication regarding a human enzyme called DREC1 and DREC2. So reading through the literatures, we can find some important informations. We can find that these two enzymes have a specific structural domain. They involve with kinase activity and they localized to the nucleus of the cell, and they also play a role in the apoptosis process. So this information strongly suggests that the two enzymes, the DRECs, are the integral components of the intracellular transduction, signal transduction. So the experts will transform this information into a structured database records called gene ontology annotation. And there are three main components within a gene ontology annotations: the gene product information, the corresponding Go term, and the PMID. However, this information is very comprehensive to be extracted. And this annotation process can often induce errors. So for example, if the selected Go term is the red Go term, which is an error, is incorrectly selected as a positive regulation, then this will be considered as a inconsistencies between the record and the literature evidence. And obviously, this is quite challenging to human curators when doing the manual annotations. So since 2018, the database community has placed a significant emphasis on prioritizing the task of database quality assurance. They focus on constantly reviewing the quality of existing annotations. However, the current quality assurance approaches have notable limitations. So this method is they're time-consuming. They heavily rely on labor-intensive manual literature review. They necessitate curators with domain expertise. They require the construction and constantly revision of comprehensive guidelines. And the fourth point, although there have been attempts to develop automatic or semi-automated systems to assist this curation process or to accelerate this process, they operate within a predetermined context, lacking the ability to leverage broader evidence, especially like the relevant domain-specific background knowledge, which I would introduce later. So if we narrow down the focus to the gene ontology annotation resources, we can also notice that this manual quality assurance cannot scale up with the growth of the annotation. So on the left indicates the accumulation of the annotated publications that needed to be constantly monitored. And on the right shows the current total count of the literature-based Go annotation with a property assigned as exp. And another thing is generative large language models. So these models, they can process the text, but they're not reliable for detecting the inconsistencies because they're prone to hallucinations. Like this example shows, providing the snippets of literature evidence, and we ask the questions because the snippet says it may have a role in the cytokinesis. It has not been confirmed yet, but if we look at the answer of the large language models, it gives a definitive absolute answer, which can be incorrect. And many research have reported that these large language models cannot meet the medical grade standards. So to tackle the introduced challenges, we developed a feasible prototype automatic systems to detect these inconsistencies. And most importantly is we have to ensure that the system can satisfy a human-in-the-loop or AI-in-the-loop application setting. So our studies involves with the answering of 4 major questions. I'm not sure why the animation of my slide is not working. So it only shows the 4th questions. So firstly, the first question is we found the Go annotation inconsistencies, which can be considered as a record level inconsistency, are very prevalent in the databases, but have not been well studied. So in our paper, we defined the topology of 4 most prevalent inconsistencies. Secondly is because there is no existing framework for automating the annotation inconsistency detection, so we proposed a solution by formulating the detection of inconsistency as a text classification task and designed an automatic framework that can suit the AI-in-the-loop application setting. And thirdly, because we are aware of that external biological background knowledge are essential to support the evaluation of the consistency of Go annotation. So we explored various kinds of biological background knowledge and proposed a feasible approach to integrate them as part of the inconsistency classification. And lastly, which is also very important, is we propose various approaches to evaluate the reliability of our designed inconsistency detection system. And we evaluate its performance under both general and several comprehensive special scenarios that can be encountered in the practical settings. For example, if we are training the model using common species data and we tested rare species, we can notice that the performance sometimes can drop very significantly. So let's dive deeper into the details. Defining the topology of inconsistencies is the first challenge we face. So first of all, it is impractical to manually outsource various occurrence of inconsistencies from the database because it is costly and time consuming. And secondly, it is not feasible to find inconsistent annotations by tracing their revision history because most of the biological databases only provide public access to its most up-to-date version. So as a solution, we opt to define inconsistencies as an instance that violate specific curation guidelines. And in total, we define 4 types of inconsistency. The definition of these 4 inconsistencies are well accepted by expert curators, and I would introduce one type of this inconsistency in detail, which is the over-specific selection of Go term. So the over-specific selection of Go term occurs when a selected Go term provides more detailed information, which is not fully supported by the literature evidence. So pulling out an example of annotation on the left and a snapshot of the Go hierarchy where the selected Go term suckling behavior located at on the right. So we can clearly find that this Go term is too specific. The evidence does not specify that the intake of food is conducted by taking the nourishment from the breast. So a consistent annotation shall be made with a selection of a broader Go term, which shall be the feeding behavior. So this example sort of indicates a challenge that is commonly faced by human curators. So based on the definition of the typed inconsistency, we can simulate real-world inconsistent instances to support the implementation of statistical inconsistency detection model. Here I will introduce the heuristic strategy to simulate over-specific inconsistency. So this strategy involves transforming a consistent instance into a specific type of inconsistency. So firstly, which is on the first line, we randomly sample a manually reviewed and experimentally validated instance from the biological databases. These instances are considered the most reliable resources in the database, assumed to be consistent. And next, referring to the Ontology hierarchy, which is on the second row, we randomly select a child term to replace with the original Go term. And then the new Go annotation will be a valid over specific type inconsistency record. And moving forward to the inconsistency detection framework. In short, the proposed framework takes each Go annotation instance as the input, extract and process the key information such as the literature abstract, the gene information, the Go terms, and then classify this information, this instance, into a typed inconsistency which is defined. And after the classification, the flagged suspicious record will be sent to human curators for further review. So, and in total, this formulates a human-in-the-loop framework. And you may speculate that one of the assumptions that we applied here is the multi-type single-class classification setting. It is not the actual case in the Go annotation, because an annotation instance may be more than one type of inconsistency, while this setting assumes that each annotation can have only one type of inconsistency. So this is a very potent speculation, but this assumption is still feasible when it is applied in a real-world application setting, because this framework is designed to satisfy a human-in-the-loop application. So if a suspicious instance can be classified by the automatic systems, regardless of the number of inconsistencies it might involve with, this instance will still be forwarded to human curators for further review. And another advantage of this assumption is it inherently entails ranking mechanisms, because it is trying to classify the most likely type of inconsistency an instance can involve with. Apart from processing of direct information in the Go annotation, the reference of related biological background knowledge is also important to support the detection of inconsistency. So there are some existing studies that rely on such external knowledge to ensure the quality. But the problem is there has been a lack of systematic studies exploring the diverse type of background knowledge. And also, there is no study in trying to answering how this kind of information can be specifically integrated to detect a literature-based inconsistency. So in our study, we conducted a systematic investigation towards the utilization of background knowledge. These are formed in 4 categories, but today I will typically introduce one of them, which is the existing annotation pattern or annotation co-occurrence. So the annotation co-occurrence refers to a group of gene ontology terms that are collectively assigned to the same gene product. From a biological perspective, genes that are of the same family or exhibiting similar functions tend to have shared annotations. And these patterns can aid in detecting inconsistencies or detecting outliers in the geontology annotation. So as illustrated in this diagram, the Go term antigen processing, which is the third Go term, antigen processing and presentation is more likely to be associated with the red gene, as opposed to the fourth Go term, which is the T cell activation. So in order to encode this information, we propose transfer learning. So we use transfer learning to encode the annotation patterns and integrate them into the automatic inconsistency detection. We model existing annotation as undirected heterogeneous graph. The terminology heterogeneous is because the nodes that we're modeling are of different types, which are gene and Go terms. And we design a graph neural network model with the objective of edge type classification to encode the annotation patterns as node embeddings, predicting whether the edge annotate or not annotate between the nodes. So this is the structure of this graph neural network models. So this diagram is too technical. So to simplify this, the key message is it will take interconnected Go terms and genes as the input, and then it will encode a mathematical representation for each Go term and the nodes and the genes. And then the representation will encode the information of which Go term is annotated to which gene respectively. And after the encoding, this mathematical representation displayed as H3 will be extracted to be used for combining with Bayes inconsistency detection model. And this diagram shows an example of how this information is combined with the Bayes language model. Here, I'm demonstrating using the PubMed BERT model, which is a language model. This node embedding is concatenated with the last hidden layer, and the concatenated representation is then forwarded to make a text classification. We also propose a method to modify the Bayes language model to pay more focus on the biological concept mentioned within the literature text. For example, the mentions of the genes or the mentions of the Go terms and so on. So this is achieved by introducing an entity embedding layer to encode the location of this concept recognized by the concept recognition model. And now it leads to the last research question is how to do the evaluation. So we select and designed two types of evaluation. The standard precision recall and F1 score is targeting on measuring the prediction correctness and the dataset wise, namely calculating the fraction of correct predictions against a certain amount of data. We also proposed uncertainty measure for data point wise evaluation, because a system is more reliable in predicting a specific instance if it has lower uncertainty towards its prediction. So here comes the results. So we definitely have reported like massive table of results in the paper, but for simplicity, I will only show this one diagram. It shows an abstractive visualization of the F1 score in 4 different implementations in detecting the inconsistencies. The baseline is made by simple distribution rules, which is treated as the control line. The second left bar is the implementation without the integration of background knowledge, but a basic language model. The light green bar in the middle, which is the third bar, is also a baseline, but it's for background knowledge integration. It is achieved by randomly generating embeddings with Gaussian distribution and integrated with the Bayes language model. It acts as a control line as well. And the last right dark green bar is the implementation equipped with a certain background knowledge and has given the best results among the others. The key message is by comparing with the baseline or the control line, we found it is certainly feasible to automate the detection of literature-based geontology annotation inconsistency. And on top of that, by utilizing background knowledge, can bring further improvements to that. And to gain a more comprehensive understanding of the predictions and the performance, we also conducted manual examination of the flagged suspicious instances. And here is an example. So the red text to provide the direct evidence to support the annotation of the first Go term, which is cellular osmoregulation. However, if we read the highlighted yellow text on the bottom, on the contrary, it provides speculative information. So although the cytokinase or cell division does involve protein movements, but by reading this literature, we can find this function have not been confirmed by the direct evidence findings. So the second annotation is flagged as inconsistency by the system. So in summary, in this paper, we made 4 key contributions. We defined the topology of 4 most prevalent type Go annotation inconsistency, and also proposed corresponding simulation strategies to these inconsistencies. We developed an automatic framework that can satisfy an AI in the loop application setting. We proposed the strategies to integrate external relevant biological background knowledge to further improve the detection of inconsistency. And lastly is we provide a systematic evaluation and interpretation to our implementation. And this will be the last slide of my presentation. So I'd like to send my acknowledgement to the contributors of this study. There are Dr. Benjamin Goody from the Florey Institute, Dr. Nicholas Geert from the University of Melbourne, and Dr. Karen Verspool from the RMIT University in Australia. Thank you.
Thank you. I've always wondered why the GOA, the annotation, just lists the PubMed ID. Does it really expect people to go off and read the whole paper? Like what you did there was show the section of text with the highlighted information. And it would be so nice if you could see that as a scientist and say, oh, I agree with that, or I don't agree with that. Have you talked to the GOA people about modifying what they present in the format?
No, we actually, the reason that I am displaying only the text snippet is for a more like friendly or better visualization for the presentation. In the actual practice, what we did is we are only processing the full text in the abstract of the article. The reason for that is because we have limited access to the full text, the entire articles. But you're giving a very good suggestions that yes, we need to maybe collaborate with the curators to focus on maybe highlighting the essential or key informations that tells the functions information.
So thank you. Let me introduce myself first. I'm Paul Thomas. I'm one of the PIs of the Gene Ontology project. So this, I have to say, this is really exciting to me. So I really wanted to thank you for picking up on the publication where we really wanted to stress to the community that we're very serious about QA of Go, and we think of this as really a community project. And so I just want to say, you know, we very much welcome this. I'm really excited to see this. My specific question, of course, then is, have you contacted the Go Help Desk yet? And if not, would you please? I'd love to hear from you. And so, you know, I just want to say, you know, please feel free to bring us into the loop on this. We're looking at, we're interested in working with anybody who's who also thinks this is a really interesting problem, because obviously we think, it's a very highly used resource. It needs to be as correct as we can possibly make it. And it's really what the main purpose of our project is. So thank you for taking that seriously.
Thank you.
Yeah. So have you contacted the Go Help Desk?
No, we haven't. We
right.
haven't yet. Yeah, but we're
Yeah.
definitely looking for more human-involved collaboration in future.
Yeah.
So to further improve the entire design of this system.
Yeah. Excellent. We'd be happy to help out. Thank you.
Thank you.
So I'm actually curious about your definition of inconsistency. And I guess because humans wrote some text, right? And even though maybe it wasn't supported fully by evidence, they still thought it was worthy of mentioning that in the abstract, right? And so do you think there's maybe another category you can consider, which is kind of verified truth of evidence, right? Something in the middle, and then inconsistency or error? Would you maybe think of a different approach there a little bit with one more level of classification, right?
Yes, you're right. Yeah, so I think the current issue is, which is also one of the key challenges for this study is, if we really want to have a better categorization of the inconsistency, we definitely need human involvement in, for example, really looking at a certain amount of data and trying to pull out these inconsistencies from the huge pool of the data, and then we sort of start to characterizing this type of inconsistency. But it sounds very costly and time-consuming. So as the initial feasibility studies, we opted to, so let's focus on the most prevalent type, and we assume that those that seriously violating the curation guidelines, or QA guidelines, are the primary types that we need to look at.
Okay, thank you. Fair enough.
Thank you.
I'm just curious. I'm with the whole human in the loop thing. I'm worried about how many of these annotations are you going to be flagging falsely for humans to have to look at? I'm thinking of cases like where maybe the abstract doesn't have the more specific information, but it's in the body of the paper, and then the human has to go and read the paper and figure out whether. I'm just wondering how often this is likely to happen, and is it actually going to save time?
Yeah, that's a very good question. So I think it also has one of the evaluation metric to answer your question is we have precision-oriented and recall-oriented evaluation to sort of balance. We definitely don't want to achieve very high recall, like flagging a large amount of data to increase the human powers again, human efforts again. But I think it really depends on which collection of data that we're looking at. If we are applying the systems to detect inconsistencies on very common species like humans or mouse, they can achieve a quite good performance. But if we apply this to some rare species, then there are lots of problems and challenges. It may significantly increase human efforts in reviewing the flagged suspicious records. Yes. Thank you.
I have kind of a related questions. Yeah, I was wondering how fine-tuned this is to go, and how you think can be, generalized or even applied to another ontology annotation.
Well, it's a very big question. Yeah, we haven't really actually looking at other ontologies. Yeah, but we will take a look. But yeah, I don't know how to give you the answers now.